Yeah,
if that's legal, right?
pretty much not.
And there's like no other countries that really have open carry, or even you can have a gun.
Yeah.
Okay, so from a couple weeks ago, we're going to review PCR and actually do the PCR with the DNA that you extracted from the different foods. The purpose of this, again, is to determine whether the food contained a genetically modified organism, whether it contains a particular gene or not. So we went over this before, but in PCR, you have several ingredients in your food. You've got your DNA, you've got your target DNA, you've got your primer, and you've got these things called DNATPs, which are just the A, T, C's, and G's. And then we have another thing which I didn't draw up here, which is the enzyme called Taq polymerase. Okay? You heat it up, 95 degrees. What happens when we heat it up?
Splits.
It gets warm and what, Brian? Splits what?
DNA.
Right. Call that denaturing. So the hydrogen bonds that hold the DNA together split. The DNA splits. Then the next cycle, and all this happens in a little machine. See that purple machine back there? All this is going to happen in that little purple machine. Then it cools, so it heats up for like 45 seconds. Happens pretty quickly. Then it's going to cool down to 75 degrees, 79 degrees. I can't remember exactly. It cools down. Then what's this? What's the green thing? Nope.
Wait, what?
It's called the primer. So the primer, again, is specific to the gene. If the gene is present, the primer will anneal. That means the primer will bond on to the DNA. If the gene is not present, then the primer won't bond, and you end up with no amplified DNA as the end result. Okay, so it cools, the primer anneals, the primer bonds on, and then it warms a little bit. You call that phase extension. During extension, you have your primer, and you also have, what color did I draw it? Blue. This is where you need the Taq polymerase, because the Taq polymerase is an enzyme that facilitates this whole process. Without the Taq polymerase, you can't have elongation. So extension, elongation, you can use those synonymously. Different sources will use extension or elongation. So what happens there is that the Taq polymerase and the primer go scooch, scooch, scooch down the DNA strand, and the letters are put in, and you end up, at the end of one cycle, you started with one strand of DNA, you end up with 2 strands of DNA. What's up?
Can I borrow some tape?
What kind?
Like the masking tape?
Yeah. If you need more, there's 27 rolls in the back.
Thanks.
Okay, so this is PCR. Again, the purpose of PCR is you have some DNA in there. We need many more copies of that DNA. What we're going to do with the amplified DNA when we have enough copies, and I'm talking like we need billions of copies of these genes, of these particular genes, you'll end up with just segments. We're going to run them through a gel, agarose gel. Agarose is derived from seaweed. So if you visualize seaweed, that's like not a bad analogy. So it's kind of like netting, it's like getting through. So you run the DNA through this agarose gel, and the small strands go through more easily, so they'll travel farther, faster, and the long strands get caught up. So you'll end up with, where do I want to draw this? And that lesson that we did at the end of last week, you were supposed, I don't know, I think you got this far. You'll end up with an agarose gel that has a banding pattern, that has a DNA fingerprint that you're kind of familiar with, and you hear DNA fingerprint on CSI and whatnot. In an agarose gel, usually you have one lane that uses what's called a marker, that uses DNA strands of a known length. It's like a ruler. So you get that from the company, okay? You don't have to create that, or all that comes, you order this stuff in kits. So you put the DNA marker in there. So these are bands of known lengths. You use those as reference points. And then you put your unknowns in the other lanes, and wherever the bands end up, then that will help you to identify what genes are present. So then you look at the banding pattern, and you have to draw some conclusions about, okay, that band means that the GMO gene is there, or it means that it's not there, or, okay, and stuff like that. So the protocol to do this is fairly intricate and involved, and it's important that you do all the steps really carefully. So I'm gonna have you in your lab notebook make a visual flow chart of this procedure before we start it. So this is the PLTW lesson. In this activity, you'll complete a lab procedure to prepare the DNA. You'll set up and run the PCR reaction specifically designated to detect the presence of the 35S promoter and terminator genes. So those are genes that are in 85% of all GMO foods. And we'll circle back around and come and look at what those genes actually mean a little bit later. And by later, I mean like tomorrow. So it gives you a big long list of your equipment and then your procedure. In this part of the investigation, you will set up 2 different PCR reactions for each of the 3 DNA samples. Test food, non-GMO food control. Do you remember those lists of foods on the back? Do you remember which one was the non-GMO food control?
The wheat.
Nope. Oatmeal. Yeah. Call it. Actually, I knew what you were talking about. Yeah. So the oatmeal, that's your non-GMO. So we should have 2 samples of that. And then your GMO positive control, that comes in the kit. So the other samples are number 1 test food. So we have 6 of those. What do we have? How many I had each of you do? There was 4 of you.
Each of
Yeah.
us did 2.
Okay.
What they thought.
I thought we'd have another student, but that's fine. Okay. So go ahead and jump ahead to the next page where it says number 3. Turn the page over. Number 3. Select all the equipment needed for this part of the lab. So the ice bath is that white styrofoam thing. The GMO master mix, I got all the stuff out of the freezer. It's all here. The PCR tubes and whatnot are in this box. Little floaties. Some of these are screw tops, some of them are flip tops. There's a time and place for each. So what I would like you to do is in your notebook, make a little flow chart, something that looks kind of like that, for steps 5 through 10. Steps 5 through 10. So you need to know, you need to have a plan. You need to know what you're going to do and what you're going to use. And when you start reading all these steps, 5, 6, it kind of makes your eyeballs roll back in your head and you're like, what am I doing? So if you make a little visual of it, sometimes that helps your brain process it. So full flow chart, something that looks something kind of like this. Like this. That's what you're after.
Brian, you're gonna end up putting gloves on. So don't wash your hands, that doesn't apply steps.
He's probably just washing his hands anyway, and you need to have really good aseptic technique,
You're gonna put
like
gloves
not dropping the pipette tips on the floor.
on. That's because they didn't have top.
Look up,
on this bathroom.
That.
It was just sitting on the bar.
Put this stuff in the middle of the table, please. Maybe that will help. I'm very visual. When it
till